Hello all. Today we'll be starting with our first chapter, and in this chapter we'll be starting with the anatomy first. So first of all, let me tell you what anatomy is actually. Anatomy is the study of internal structure. Before coming to the other processes, before coming to study about the other processes in a plant, we must know what actually the structure of a plant is. Fine, we must be knowing about the internal structure of a plant. So we'll be studying with the anatomy first. So to study the anatomy, we'll be studying about the tissues, about the plant tissues. Fine, what is a tissue? So let me tell you, a tissue is a collection of cells performing a specific function and which have a common origin. These cells in a tissue, they have a common origin. And you can say a plant tissue is defined as a functional unit connecting all organs of a plant. So let me tell you here, the plant tissues, these are broadly divided into two main types. Number one is meristematic tissue and number second is permanent tissue. So first of all, we'll be studying about the meristematic tissue. So let me tell you, meristematic tissue, you must have already heard that it consists of a group of cells that have the ability to divide. Fine, these meristematic cells, they have the ability to divide. And let me tell you, these tissues are made up of small cuboidal cells which are densely packed. Here you see, if you observe a meristematic tissue under a microscope, you see small densely packed. Here you see the cells are densely packed together and they are cuboidal in shape. They have a nucleus, they have prominent cell wall and they have cytoplasm. So these cells are capable of stretching, enlarging and differentiating into other types of tissues as they mature. Why they stretch? Why they enlarge? Because they have the capability to divide. So this is a meristematic tissue. Now coming to next, let me tell you, these meristematic tissues, they give rise to permanent tissues. When they differentiate, or you can say when they mature, they give rise to different types of the permanent tissues. And these meristematic tissues, they are of different types. Now let me tell you, on the basis of the position of the meristematic tissues, they may be divided into three types. Number one is apical meristem, lateral meristem, and number third is intercalary meristem. Here you see, on the basis of position I am telling, the meristematic tissue which is present on the apex of a root or a shoot is called as apical meristem. It is present on the apex of a root or a shoot. Now, number second is intercalary meristem. The meristem which is present in between the permanent tissues is called as intercalary meristem. And number third is lateral meristem. Lateral meristem, here you see, which is present on the lateral sides of a plant, and that forms the lateral meristem. I hope this is clear to you. Now coming to the second class of the tissues, that is the permanent tissues. Permanent tissues are derived from meristematic tissues actually. You see, when meristematic tissues, they divide, they differentiate, they form permanent tissues. And they have lost their ability to divide. In some cases, permanent tissues also divide, but not always. They have attained their mature form. They are further classified into two types. Now, this permanent tissue, they are further classified into two types. Number one is simple permanent tissue and number second is complex or compound permanent tissue. Here you see a brief outline of the classification I am showing you here. The simple permanent tissue, it is of two types. Number one is epidermal tissue, which forms the epidermis of various parts of a plant. For example, it forms the epidermis of leaf, it forms the epidermis of stem, it forms the epidermis of the root. This is the epidermal tissue. And number second is ground tissue. Ground tissue is simply the filling tissue. For example, cortex is there, pith is there, it is the filling tissue. Ground tissue is the filling tissue. Fine. Now this compound permanent tissue, it is also of two types and forms the vascular bundle of a plant. And in vascular bundles, it is xylem and phloem. It's only of two types, that is xylem and phloem, and together, xylem and phloem together, they form the vascular tissue or vascular bundles of a plant. I hope this is clear. Now let me tell you. First of all, if you see the structure of any root, shoot, there is epidermis first, then there is ground tissue, and then there are vascular bundles. So this ground tissue, it is further of three types. Number one is parenchyma or parenchymatous tissue. Number second is collenchyma, and number third is sclerenchyma. Fine, I hope this classification is clear to you. Now, we'll be studying about this ground tissue, and number one is parenchyma. We'll be studying about the parenchyma in detail. Parenchyma. These tissues are found in the soft parts of the plant, such as roots, stems, leaves, and flowers. The cells of this tissue are loosely packed and contain large intercellular spaces between them. Here you see the cells of a parenchyma. They are found in the plant where the soft tissue is, like in the roots, stems, flowers. And this tissue, the cells of this tissue, they are loosely packed. They are loosely packed together and they have large intercellular spaces. Here you see the intercellular spaces are large. Each cell has a vacuole. Here you can see the vacuole also. They have a vacuole at the center, and the function of the parenchyma tissues are storage, photosynthesis, and help the plant to float on water. So this is all about the parenchyma tissue. Here is a transverse section and here is LS longitudinal section. How they appear in the longitudinal section and how they appear in a transverse section. But please remember, the cells in a parenchyma tissue, they are loosely packed and have large intercellular spaces between them, and they have a large vacuole at the center. Now coming to another type, that is collenchyma. We are studying the ground tissue. Please remember, this is second type of the ground tissue. In plants, support tissue of the living elongated cells with irregular cell walls. Collenchyma cells have thick deposits of cellulose in their cell walls. Here you see, these are the collenchyma cells, but at the corners they have thickenings. Here you see in yellow, these are the thickenings present on the walls. Fine. So here you can see, and the thickenings, this thickening is deposit of cellulose actually. And if you see in cross section, they appear polygonal. Fine. The strength of the tissue results from these thickened cell walls. You know, they are supportive in nature, and how they give support to the plant because of these thickenings in the walls. Fine. So here I have shown you LS. Here in yellow you can see the thickened corners, and this is the vacuole, this is the protoplasm, and this is the cell wall. I hope this is clear. Now coming to the third type of tissue, ground tissue, that is sclerenchyma. The cells are long, narrow, thick. The cell walls are lignified with numerous perforated pits. The cells of sclerenchyma are dead and without protoplasts. You know, actually these sclerenchyma, their cells are dead and they don't have any protoplasts. Fine. Sclerenchyma may be in the form of a fiber. You can say sclerenchyma cells, there are further two types. One is fiber and number second is scleroid. Fiber, you can say it is long, elongated. Fine, it's a long, elongated, it's also dead and it has a lumen. Fine, the protoplast is not there, only it has a lumen there and it has thick wall. The fibers are thick walled, elongated and have pointed ends. Now coming to scleroid are spherical, oval or cylindrical. Fine, and here you see these are the perforations, and these perforations are called as pits. These are called as pits. Pits are also present in a scleroid. Fine, so this is all about sclerenchyma. So today we have studied the tissues in the plant. Please go through the lecture and let me tell if there are any comments or queries. Thank you.